Okay, in chapter 16, we're moving on to mutation, repair, and recombination. This may be of particular interest to those of you that are going into the medical field because, as you know, mutation repair, cancer is a big problem when you're not able to repair these mutations that occur in DNA, and that can lead to tumorigenesis and things of that nature. Specifically, some of the well-known ones are BRCA1 and BRCA2, the breast cancer genes that kind of stop this mutation repair mechanism from being able to get rid of cells that have incurred these poor mutations, which leads to tumorigenesis. So here's the chapter outline, as always, just for your reference. So as we covered previously, there are consequences of having point mutations within a gene. In the earlier chapters, we talked about the idea of codons and frame shift mutations, where an insertion or a deletion will cause the entire codon sequence further down from that mutation point to be shifted, and thus all those amino acids are shifted. However, if we have an insertion or a deletion in groups of 3, which represents one codon, that causes a change to that one amino acid, but the frame stays intact. And so if we look at this example here, we have a sequence where we have our 4 amino acids that are generated from these 4 nucleotides. And then let's look at an example here of a transition or a transversion. So at the C in the arginine, we have a change, or sorry, at the A, we have a change to a C, to a cytosine. And that causes this synonymous mutation where we change the codon, but that makes the same amino acid. And so this is kind of a silent mutation. And remember, when we talked about these codons earlier, we talked about how there are multiple codons for each amino acid. And usually the first 2 amino acids of the codon, for in this example A and G, those are pretty well conserved. But then that third codon is what we would call a wobble base. And this base can vary and still code for the same amino acid. And so if we have a change, a point mutation in that wobble base, generally a lot of times we'll end up with the same amino acid. Next, we have a missense mutation. And so this is a mutation, as you can see, we have a G that turns to an A, and thus an arginine turns into a lysine. And so this is an example of a conversion where the codon that we have switched to from arginine to lysine specifies a amino acid that's very similar chemically. And so thus a lot of these mutations, these conservative missense mutations, they might have kind of a leaky function where they have reduced function, but generally there's not a complete destruction of that enzyme from this mutation. And so we might have, like I said, poor function, leaky function, but because those 2 amino acids are so similar, we're generally going to not see much of an effect phenotypically from this mutation. And so now we have a missense mutation that's non-conservative. And in this example, the altered codon specifies a chemically dissimilar amino acid. And so this causes a little more chaos in terms of how the resulting protein is affected, because since these amino acids are no longer similar, there's different binding and different folding of that protein, so we have changes in the tertiary and quaternary structure potentially that will cause this enzyme to no longer have the function that it previously had. And then lastly, we have this chain termination. So this is a nonsense mutation. So this is an example where we have a mutation. So in this lysine over here, we have a that is now turned to a T, and what that has done is caused that second lysine to instead become a stop codon. So what this does is it truncates that entire amino acid sequence, and so we have kind of a nubby little amino acid nubby little protein that's only the first couple amino acids instead of that entire long chain. And thus, this really has an effect because who knows, down here and this glycine down at the end may have been the a binding point for this protein, or the functional point of this protein, and it's no longer there because the ribosome, as it's reading this mRNA, it sees the stop codon and it says, okay, I'm done making amino acids, and so the protein no longer functions. So for these last two types of mutations, we're going to talk about indels. And indel stands for insertion or deletion, so it's the first couple letters of each of those words. And so first we'll talk about insertions. And so an insertion here we have this G that is inserted after our first codon. And so thus every codon after that first G is shifted down, right? And so there's now a change to all three of those amino acids after this initial amino acid. And so we call this a frame shift mutation, and it really changes the function of the protein because you're not just changing a single amino acid here or there within the sequence, you're changing everything after that initial insertion or deletion. And then lastly, we have a frame shift mutation for base deletion, or the del of indel. And here you see we have where our A used to be, we have deleted it, and it causes all these amino acids to shift forward one, which causes our codons to be shifted. And so just like an insertion where you're shifting everything to the right, with a deletion you're shifting everything to the left, and you still have this frame shift where the rest of your protein sequence is all messed up. Another example that we had very early in the course of a damaging point mutation that is not an indel is when this point mutation happens at an mRNA splicing site. Remember, we have this AGGU rule that tells us the boundaries of these introns and exons. Now, that spliceosome that is hooked to that carboxy tail of this transcription machinery, now, it's looking for that AGGU. So if we end up having a point mutation at one of these nucleotides, or conversely on the first boundary here, or on that conserved A branch point site, those are the three spots that were really important for the spliceosome to recognize where to splice out introns. Now, if there's a mutation there that changes those away from an AGGU or this conserved A, then we're in trouble, right? Because that spliceosome no longer is going to recognize that there is a splice site there. And so when the amino acid sequence is then translated or translated from the mRNA, this intron, which is not supposed to be there, is still going to be incorporated and read as though it's an amino acid sequence and not spliced out prior to the mRNA being created. And thus we have a protein that has this big insertion in it. And if we remember our example with the neomycin insertion in the mouse, just inserting a big chunk of a functional protein, or even in this case a non-functional intron, into a protein is enough to mutate it and break it. So this mutation, point mutation at this AGGU site, or that conserved adenine, is really damaging to the protein and can cause a lot of harm in a big mutation. So now we're going to look at these consequences of mutations kind of in a more holistic way, looking at both the mRNA, the protein, the gene, and then how they would track on a gel, both a northern blot, so looking at the actual mRNA, and a western blot looking at the protein. And so with a wild-type gene, we have a normal length mRNA, normal length protein. We have a mRNA that tracks, this is our kind of control for the northern blot, and then our control for the western blot. Now if we have a missense mutation, so we're changing one of those nucleotides, and a lot of times, remember, this is not as damaging as a frame shift, we see that we have a kind of explosion graphic here that shows that we have a mutation in one of these amino acids. That mutation is then transcribed into mRNA, where it is also that nucleotide is mutated in the mRNA. And then we have a protein who we have one amino acid that's changed. Remember, these missense mutations are changing just a single amino acid, thus it is changing a single codon. So we have a protein that is slightly different because it has one amino acid that has been changed. And so if we were to blot these, remember these blots, they segregate stuff based on size. And so the mRNA is still just as long as it was in the control. And the protein, that protein is also just as long as the control because we're not gaining or losing any amino acids. We are just changing a single amino acid. Now let's look at a nonsense mutation. So remember, a nonsense mutation is when we change an existing codon that reads for an amino acid to a codon that instead reads for a stop codon. And so again, on the DNA, we have this explosion that represents our change of a C to a T. The gene is still transcribed, right? And so this stop codon isn't telling the transcription to stop, it's telling the translation, so the ribosome, to stop. So the gene is still transcribed fully in terms of the mRNA. Then, when we get to the ribosome and it's reading those 3 codon triplets, it will read this mRNA until it gets to the stop codon. And so this new stop codon causes the protein to be truncated or shorter. And so if we were to run this example on a blot where we run the mRNA, we see that, remember, the mRNA, that stop codon is not telling the transcription to stop, so the mRNA is the same length as it would be in the control down here. However, if we were to look at the protein is instead truncated. And so on these gels, remember, smaller things run further, they run faster because it's kind of if you're big and clunky, it takes more time for you to get through the gel, so small things go through quicker. And so from this band here, we can see that the protein is shorter than it was in the control over here. So now we're going to look at a frame shift mutation. And so in this case, remember, frame shift mutations can arise from either an insertion or a deletion, those indels. And so in this case, we're going to have an insertion of an A. And here we have where our event happens, the same place, relative in this gene. And if we look at the mRNA, everything downstream of this insertion is going to be changed because it causes every amino acid to be shifted down one, so then your codons, your reading frame, is shifted. So if you remember our example from earlier in the course, we had the fat cat ate the big brown, or the fat cat ate the big rat, and if you have an insertion, you get the fat cat garbly, 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 because we had everything that got shifted downwards. And so this is kind of conceptually what we have happening here. And so we have our mRNA that's changed, and we have our protein where that has also changed, and every amino acid downstream of our insertion site has changed. And so if we were to put these on gels, we would see that in the case of the mRNA, we have really not a detectable change. It's really, really hard to see a single base pair difference in mRNA if you were to run it on a gel. You'd need a tremendously long gel to decipher the difference between, say, 250 and 251 amino acids. So for all intents and purposes, this amino acid or this mRNA running on the gel is the same as control. And the same thing with the deletion. If you went down from 250 to 249 amino acids in an mRNA strand, you're not going to be able to resolve that on a gel. It's not that specific. However, let's look at proteins. So what's interesting here is you might notice that it says we can either have shorter or longer proteins as a result of this frame shift mutation. And so think about that for a minute. How is it that we can get either longer or either shorter proteins because of a frame shift? Well, you have to remember that the way that the transcription machinery, or the translation machinery stops a protein is with those stop codons. And those stop codons, that codon is shifted as well. And so if you shift the stop codon at the very end of this amino acid sequence, it's possible that this amino acid sequence will run on a lot longer than normally did. And thus, we would have a band up here where we would have a much bigger protein. But conversely, what could happen is this frame shift could cause a stop codon to appear prematurely. So I should probably put it in the right spot here. So it could happen much earlier than before, just by random chance of shifting those amino acids by one. You could end up with a TAA that wasn't there before. And so in that case, you would have a protein that is truncated and shorter. So it's all random chance, depending on the sequence, that you could either get rid of the stop codon and it would go on longer, or that frame shift could create a premature stop codon and you get a much smaller protein. I really like this last example because it's really easy. So what if we have a mutation in a regulatory region? So for example, the TATA box, or that -35 region in eukaryotes, or sorry, in prokaryotes, or transcription binding site, or something like that. So if that's the case, and that transcription machinery is not able to come in and actually transcribe the gene because they've kind of lost the ability to figure out where that gene is, or the ability of that polymerase to bind, or something of that nature, what happens is we don't even get mRNA, because that mutation is so damaging that, or so inhibitive of that gene, it doesn't even allow mRNA to be created. And thus, if there's no mRNA, the translation machinery, the ribosome, that's not able to read anything because no message ever made it there. And if we were to try to run this on a gel, we just have nothing, right? Because we had no mRNA. Since there's no mRNA, which are the blueprints, there's no protein, and thus our gel would just be empty. Okay, let's look at how these indel mutations occur generally. And so a lot of times these indel mutations occur because of repetitive regions of DNA. And so during synthesis, we have this kind of shifting. And so here we have a blue template strand, and this yellow strand here is the strand that's being synthesized. And as you can see, on the blue strand, we have a large swath of repetitive nucleotides. So as the strand is being synthesized, we get this slip. And so this T kind of gets pushed off into a bubble. And then because there's still more complementary bases, these newly synthesized T's are allowed to still match up properly. And so the result of these ATs still being paired together kind of holds this bubble in place, right? And so then that strand finishes, and as you can see, the newly synthesized strand has this additional T that's been inserted into it. And so during the next round of replication, remember both of these, both the yellow and the blue strand, are going to be templates for the next round of replication. And so when this yellow strand here is transcribed, or sorry, is replicated, we have now this T that is no longer in a bubble as it gets pulled apart, that is able to act as the template for this new strand, this blue strand for replication, and it will incorporate that new T. So deletions of base pair happen in a very similar manner. So in this case, instead of repetitive sequences of just A's, we have a repetitive sequence of CTs. And as this synthesis is happening, this shift occurs, in this case, in a deletion in the template strand. And so the newly synthesized strand up here in yellow, that is chugging along just fine, but the template strand has slipped and caused this bubble. And because there's those repetitive CTs and their paired GAs, those are able to kind of still hold everything in place, right? Because they're forming their normal bonds, and that overpowers that CT's ability to look and find a bond of its own. And so then, as the sequence is completed, it stabilizes, and we have these two matched strands of DNA where the original template has the bubble, but those two strands are then used for the next round of replication. And so when they split apart, the template is going to look very different, right? And so the top template here is only containing 4 of those GA repeats, right? And so down here, that template is used to create this blue strand. And as you can see, these daughter cells of the, from the process of replication of the genome only has 4 copies of that GA or CT. 1, 2, 3, 4. Now, this original strand that had the bubble, it goes back to its replication, and as you can see, it still has that original CT. It was just in a bubble and kind of misconstrued, and so it, during replication, has all 5 of those original CT pairs. And so in this cell that used that bubbled one, we have no deletion, but in the synthesized strand that did not have the bubble, we see a deletion of those CT. So earlier in the course, we also talked about these microsatellites and mini satellites and these conserved nucleotide repeats that are present in DNA. And so a lot of these repeats can be used for genotyping individuals or determining who a different individual is. This is actually used to be, I don't know if it is anymore, but it used to be a way that forensic science was done. So you could find a gene that has variable repeats between people and then do PCR or some other method to actually count the number of repeats this individual has, and that would be a way of actually kind of fingerprinting somebody because different genes have different number of repeats, and this is specific to different individuals. And so microsatellites and mini satellites were used for this. Now with the onset of next-gen sequencing and SNPs, single nucleotide polymorphisms, that may be a little more accurate, and they may have moved to that technology, but this used to be a big thing for genotyping, and it's still used in organisms that don't have sequenced genomes, very novel organisms or understudied organisms can still use a lot of these markers. And so the reason that these different individuals have different number of these repeats is because of this repetitive sequence, just like we saw previously. So similar to how we get that deletion, those indels, insertions or deletions, those bubbles that form during the replication process, we can also have this happen with these nucleotide repeats. So here we have a trinucleotide repeat, which is CAG, and then the N represents the variable number of these repeats depending on the individual. And so during the process of replication, we can get this slipping that we saw previously, where we get the formation of this bubble, and because these repeats are able to bind to themselves, so CAG will bind to GAC, right, backwards, and so we can form these loops where they're kind of willing to bind to each other, and at that point it's kind of protected and it's comfortable being in this loop, and so during the next process of replication, the daughter strand will have an additional 4. See, there's 4 boxes here. 1, 2, 3, 4. We'll have an additional 4 of those trinucleotide repeats. And so it doesn't happen every generation. So usually when you do genotyping on like family trees, you will have very similar nucleotide repeats to your parents. But it does happen once in a while, and it depends also on the gene, how common the slippages are. Some genes have more variable numbers than others, some are more conserved. But just like the mutation, the indels we saw, this same kind of bubble formation is what leads to this very copy number in different individuals. So now we're going to talk a little bit about some of these mutations to base pairs as opposed to slippage, how like UV light and some other mutation-causing components can change DNA. And so UV light can cause formation of cross-links that are not supposed to be present in DNA. So you can get cyclobutane pyrimidine dimers that form. So this is kind of a cross-link of two pyrimidines within the DNA chain. And you can also get these photoproducts where one of the bases will form a bond with this double bonded oxygen with the next base up. And so when that happens, so we kind of have this cycle. So normally this is what our DNA looks like. We have our two base pairs in here with no mutation, but UV light is exposed to the DNA. And at that point, we get this photodimer, so we get the two bases bound to each other as opposed to binding across the strand to their complementary base pairs. And so through evolution, organisms have found ways to combat this, because if you mess up your DNA, you're messing up your ability to reproduce and carry that information to the next generation. So it's very important. We also talked about how DNA is the most stable form of this information of a gene. mRNA is much more vulnerable, single-stranded. It doesn't last long. It naturally degrades after time, and proteins aren't able to replicate themselves. So DNA is kind of the blueprint, and that's very important to keep that blueprint intact. So when we get these photodimers that are created, we have different enzymes. In this case, we have CPD photolyase, and what that does is it comes in, and when it's exposed, this enzyme is exposed to light greater than 300 nanometers, so that's the wavelength. What it does is it'll come in and it will repair and break apart those two photodimers, and then we're back to normal. And so this process can happen over and over. This is just showing that it can happen over and over. It doesn't keep going in the cycle continuously, but this is one of the repair mechanisms. Okay, now let's look at how minor base damage is repaired through this base-excision repair mechanism in organisms. And so in this example, we have a base indicated in blue here that is damaged, so it's not correct, not the proper base. So in comes an enzyme called DNA glycosylase, and what that does is it cleaves the actual base from that sugar backbone. So the actual nucleotides removed from, remember, the three parts of DNA. One was the sugar backbone. The base is removed from that. Next, AP endonuclease comes in, and it makes a cut in that backbone where the base was attached. From there, DRPase comes in, and what that does is it uses that little cut, and it will remove a bunch of additional nucleotides upstream from where that nick was made from AP endonuclease. Then polymerase can come in, and it will find that site that is missing. It's only single-stranded, and it can use the upstream site for kind of the primer that's required to get that polymerase onto the DNA, and it will synthesize this new strand here. And then finally, remember, just like in the replication component of this chapter, we end with this synthesis part with a nick in the DNA still. Remember, the polymerase can't fill in all the way on those lagging strands for DNA replication. Similarly here with base-excision repair, it can't fill in that sugar backbone. So that's the role of ligase once again. Ligase comes in, it fills in that nicked backbone, and now we've completely not only fixed that one base pair, but also some of its neighbors. So what happens when we have a change of a nucleotide, like a point mutation, as opposed to just a damaged base? And so there are two pathways for the excision and repair of a base, that point mutation that has resulted in a mismatched base. And so as you can see here, it is kind of represented by this mismatched base, this point mutation. Because that base is no longer with its proper pair, there's going to be a bubble, right? So if this was originally an AT, if it turns to a G, they're not going to be able to bond together because they're only looking for their complement. So you get this kind of bubble formation. And so the two pathways that are responsible for fixing these bubbles, or these nucleotide excision repair are global genomic nucleotide excision repair, or GGNER, and the other one is transcription coupled NER, so nucleotide excision repair, or TCNER. So we're going to go step by step over each of these two repair pathways. So in GGNER, these bubbles formed by improper base pairing from a point mutation are recognized by this XPC-RAD23B. Now don't worry, you don't have to memorize that whole complex or long name for this, but just realize that there is an enzyme that recognizes and binds to these bubbles formed from improper base pair. In the TCNER, polymerase, RNA polymerase, so remember RNA polymerase is transcribing this gene, and a mutation could be recognized to a gene that's already been transcribed and not going through replication. So RNA polymerase is going to run into this bubble when it's transcribing a gene, and it's going to stall because there is a problem where there's kind of a bump in the railroad track, so to speak. And so RNA polymerase is going to stop and be kind of hung up on that bubble. Now here we have our two nucleotide excision repair pathways combining. So the first part is kind of the recognition step. In step 2, the DNA damage recognition complexes, which is this XPC-RAD23B or this polymerase, they will recruit this transcription factor 2H, TF2H, to that bubble, that nucleotide that is mismatched. Here, XPB and XPD are recruited by that transcription factor 2H, and they will bind outside of this transcription factor machinery that has recognized this lesion and kind of act as sort of an endonuclease, and they will excise the damaged DNA segment. So they come in to the boundaries outside of the lesion, and they will cut one strand of that DNA and get rid of this damaged segment of DNA, as well as some base pairs flanking it upstream and downstream of that lesion. From here, DNA polymerase comes in once again because we have a single-stranded piece of DNA because we took that chunk out that had the lesion in it. And it will use these upstream regions as a primer that it can kind of bind to the double-stranded DNA. And it will fill in this hole that was created by XPF and XPG. And using the complementary strands down here as a template, it will repair and fix that string of DNA that had the lesion or the bubble in it. And our end product is a strand. Again, it's kind of not very clear on the slide, but there will be a little gap here, and the final step of this is that a ligase comes in and ligates that final strand, the sugar backbone, so we have a completed, fully intact piece of DNA. Sometimes during the process of replication, we have errors that the polymerase makes. And so as this replication is happening, sometimes they'll accidentally incorporate the improper base pair, right? So we have a G and a T paired with it here instead of a C, so that's not correct, right? And so replication is a very high fidelity process, so very rarely does the wrong base pair get matched up. But when it does, we have a mechanism to go back and fix that. And so as this replication fork is proceeding, there is this enzyme called mutase, and it will kind of follow along to see if there's any of these mismatches, because we'll get those bubbles to form again. And it sees here that we have a G and a T, and so that's a bubble formed. It recognizes that and says, this is not correct. And at that point, the mutase has to look at this G and T and say, okay, which one was the parent strand and which one is the improper newly synthesized strand. And so what it can do with its little buddies, mute H, and mute L, which kind of form this complex, it'll look along the DNA strand to see if there's any methyl sites. Remember, these methyl groups are added to genes as a kind of a way of epigenetic modification. So it's tags that are put on genes to kind of turn them on, turn them off. And so what's interesting about methylation is it happens much later after replication. It doesn't happen immediately when a new strand of DNA is synthesized. It takes a little bit of time. And so if we look near the replication fork, the only strand that should have these methyl groups on it was the original template strand, because it's existed long enough to accrue these methyl groups. And so mute H will then bind to the methyl group, and then mute L will kind of link that original mute S detector, the one that's bound to the bubble. It'll link it to mute H and kind of form this complex. And then it'll nick the daughter strand and kind of start to cut out the DNA where it may have messed up. And it'll be excised, and a polymerase will come in and it will fill in that strand, including that bubble-causing base. So that base that was originally a T is now a C. And so this kind of protects the fidelity and makes sure that the replication process is very good at its job and doesn't incorporate a lot of mutations. Okay, so we've seen how mismatch repair occurs after replication, but what happens if the replication machinery is churning along the single-stranded DNA, using that as a template for replication, and it runs into DNA damage, whether that be a mutation or a lesion or some sort of other damage. So what occurs is DNA polymerase III will be using that template strand, and when it runs into this lesion or this mutated inappropriate piece of DNA, it will stall at that point, and these little proteins called Rec-A will come in, and they will bind to kind of preserve that single-stranded DNA from being attacked or binding to other things, kind of like we saw with those single-stranded binding proteins previously. Then at this point, DNA polymerase is removed from the single strand, and the beta clamp still stays there, but instead polymerase 5, or also known as bypass polymerase, will come in, and it will attach to the clamp and take the place of polymerase 3. And what bypass polymerase does is it's a little more liberal with what it uses as a template strand and what it does when it sees lesions and things of that nature. And so bypass polymerase will continue to just go right over that mutation or that lesion in DNA, and will continue to synthesize this double DNA strand. And this proceeds for a couple dozen base pairs, and then polymerase's job is done. So it's not a very high fidelity long-term polymerase. It just comes in, says, I'll take care of doing the synthesis when we have a little mess up here. I'm a lot less thorough with my work, jumps over that, and then synthesizes for a little bit, falls off, and then at that point, DNA polymerase 3 will come in, reattach to that beta clamp, and continue the synthesis. So at this point, we have a lesion or a mutation that's been incorporated into this DNA strand. And so you might think, oh no, this is terrible. So a lot of times mutations are harmful, but also we need to be able to obtain or keep some mutations, because mutation is also what drives diversity. And so there are at times beneficial mutations, and mutations are what makes, changes in your DNA is what make different individuals different. And so this is something that we don't want to fix every single mutation, otherwise everyone will look exactly the same and there'll be no difference between people. At least from the genetic aspect, there are changes in expression and things like that because of environment. But for the most part, this is kind of a way that we can kind of accrue a mutation while continuing to synthesize the rest of the DNA and tolerate at least one mutation here or there. So every example of DNA damage that we've had so far are very basic, very minute or minimal damage that's done to DNA. But what if we have something like X-rays or reactive oxygen species, or something that really plays a larger role in harming DNA than just little nicks here and mismatches there? So X-rays will cause what's called double-stranded breaks in DNA. So that's both of the two strands are busted. The two parts of the chromosome aren't even connected anymore, which is a big problem for replication. And so we need a way to fix this. And as you can see, when we have this double-strand break, we no longer have some information as to the template of what is required to fill in these this missing DNA. So what happens is we have these two enzymes, Q80 and Q70, that come in, and they recognize these double-stranded breaks with these overhangs here, and they will bind to these broken regions and these overhangs, and they will trim those overhangs back. So they will take off this little these two sections at the top here, and with the help of this enzyme XRCC4, what we'll do is use a ligase, and it will just join these two ends together. And so we've lost, in this case, probably 6 or 7 nucleotides that were in this region before. But if it comes to leaving the DNA disconnected or joining what we do have remaining back together and kind of putting a Band-Aid on it, from the organism's perspective, it's better to do that and put the Band-Aid on it, because anything that is no longer connected to the centromere during the process of replication is just going to be left out on its own, right? So it's vital, if the centromere is on this side of this chromosome, that this stuff gets reattached, or it's not going to get brought into the new daughter cells. So it's better to lose the function of that one gene, or potentially lose reduced function of that gene, or through mutation, than to just lose half of your chromosome. So this is kind of a not a great situation for the cell to be in, because you're losing a swath or a chunk of your chromosome, but it's better than the alternative. Now, not all double-strand breaks are this death sentence or this really terrible problem that can't be fixed. If a double-strand break occurs after replication, then we do have a template. It's just on another chromosome. So instead of using one of the two strands as the template, we can use that whole new chromosome as a template. So here we have a double-strand break that occurred, and we're going to trim the ends to get these sticky overhangs. So we're going to trim back on the top end and then back the other way on the bottom strand. And what this allows is for this process called strand invasion. And so what strand invasion is, those single strand or single stranded overhangs that we left is able to kind of work their way in and dislodge and bind to the complementary strand on this opposing complete chromosome. And from there, they can use, we have these double-stranded components here on both sets of the chromosomes, the blue and the red chromosomes. And as you can see, we have 2 double-stranded areas on each side with this gap here that we can use, where polymerase can use this double-stranded region kind of as a primer, and it can fill in one direction, and this polymerase up here can fill in and use that as a primer and fill in the opposite direction. And what we do is we end up with these synthesized DNAs with these what's called D loops. So they're 4 strands of DNA, 2 double-stranded chromosomes kind of connected in this D loop formation. And then the DNA will kind of unwind, and these little nicks will be filled in with a ligase like previously. I want to point out that this unwinding process, this occurs through because of like homologous recombination, so crossing over. So because the same process that allows crossing over to happen and these kind of unwind back into their or to single chromosomes, that same process is used here for repair of these double-strand breaks. So although I kind of buried the lead on the last slide, this process sounds similar to mitotic recombination, doesn't it? The crossing over that we saw in earlier chapters. Well, it is very similar. And so in this meiotic recombination, we get a double-strand break that is formed on purpose because we want to have this recombination event happening. So we have this nicking of the DNA with SPO2, and this leads to the trimming of these double-stranded because we want to get those overhangs that can invade the complementary strand or the other chromosome. And here, this part is really interesting, and there's not much known about it yet. And so these single-strand overhangs, there's these enzymes called RAD51 and DMC1 that bind to these overhangs to protect them and to help invade the complementary strands. But through some process we don't know, they're always able to find the matching strands, the complementary strand on the other chromosome. And so when we have recombination, it's always a perfect swap of one segment of DNA from your dad's chromosome for one segment of DNA from your mom's chromosome. And it's not like we swapped two different genes. It's always the same genes that get swapped. And there's not, to my knowledge, in the time of publishing this textbook, there's a lot of unknowns about how this process actually occurs. But then those RAD51 and DMC1, although we don't know how they find the perfect spot, we know that they invade this strand with those proteins and allow for crossing over to occur.